Okay, so people, I think we're about to start. If you can all move in, if you're going to move in, just to give our speakers time to be able to finish their talks. So the next session is going to be a very interesting session. It's overridden by quality at the end. And we're going to look at some of the challenges of emerging issues in stem cell processing, quality issues, and also in stem cell processing, which I think is an area we often don't think about. And then before we can even start the process, Alicia Fenter will be talking to us on Search, Match, and Connect. And she's got a title of Searching for the Most Suitable Match Donor for Your Patient, and WMDA's Search Match service. I want you to add the collective, Alicia. It comes out automatically. Just a little bit about Alicia, and then I'll just tell you a little bit about each talk and each person and presenter that will be doing the talk, and they will then sit down and be available for questions at the end. So that's the process we'll follow. So Alicia has a BSc in genetics, psychology, and human physiology. She joined the registry and worked with us in 2014, and then in 2019 started volunteering with the World Marrow Donor Association Search, Match, and Connect project, and then she transitioned full-time in 2021 as the project coordinator for the pillar and the pillar coordinator for optimizing Search, Match, and Connect. Dr. Rihanna Cochran will be doing the next talk, and that looks at challenges and emerging issues in stem cell processing, a laboratory perspective, which is something completely different for us again. And then Hiroshina, thank you for doing this. She's going to oversee everything with the quality aspect. That's Hiroshina Sengomona, and she looks after the quality and safety standards across the core divisions of Anthony Nolan: operations, patient services, the cord blood program, and H&I laboratory. She leads a small team and collaborates internally with internal and external staff on Anthony Nolan's processes and to make sure that they are fit for the UK and on international standards. She's also an assessor for the WMDA accreditation program. So those are our three speakers. Welcome, speakers, and thank you for doing your talks. Alicia, if you could come up and do your talk.
Well, good morning, everybody, and thank you so much for joining us here in the beautiful Cape Town. I really think that Cape Town is putting on its best jacket for all of our international guests and, of course, African delegates here today. It has always been my view that Cape Town is best visited in the winter time because it's nice and sunny, just a slight rain, small jacket, maybe what the Dutch would call your summer jacket would suffice. And yeah, apart from the Cape doctor treating us this morning, I think that you'll all have an excellent time here. So it's excellent that I get to speak in the quality session. While you might not think that this topic has much to do with quality, I will show you exactly why it is important to consider quality or have it in the back of your mind, even in something as routine as identifying a suitable donor for your patients in need. So, as Dr. Ingram said, thank you for the lovely introduction. My name is Alicia Fenter, and I have most of my experience as the head of patient services and senior search coordinator for the South African Bone Marrow Registry, where I facilitated a lot of the patient search, effectively at the time for all patients in South Africa. After which I then thought, well, how can we make this an even better process? How can we automate? How can we accelerate? And ideally, how can we reduce the timeline to transplantation for all patients? And that's how I ended up at the WMDA, World Marrow Donor Association, to work on the project to build out a technology that is going to facilitate that. There we go. So a quick outline on what we're going to discuss. There's also a bit of science, but I'm going to try and not bore you too much with that. We'll cover some of the bioinformatics in unrelated donor selection, as well as then the steps, the very practical steps, in how to then identify that suitable donor. So let's dive into bioinformatics, talk a little bit about the probabilistic matching algorithms that the WMDA offers in its search service, and the role haplotype frequency sets play in that. So what's bioinformatics? Bioinformatics is the use of digital tools for computation and analysis in order to capture and interpret biological data. Bioinformatics is absolutely essential for the management of data in modern biology and medicine, particularly when the data sets are as large or as complex as HLA. So how does the WMDA fit into this? Well, we develop, maintain, and support a solution called the Search and Match Service, which is the primary source of all hematopoietic stem cell donors and cord blood units globally. The database holds over 41 million records, specifically donors, and then an additional 790,000 cord blood units from 134 registries and cord blood banks across 57 countries worldwide. The Search and Match service then provides what we like to call internally the Google for donors. So you enter your patient, you hit search, and then within a few minutes, you find a list of compatible donors, irrespective of where they might be listed in the world. So how does a matching algorithm work? Well, an algorithm is simply a series of instructions that are involved, or that when followed step by step, to do something useful or solve a problem. For example, you could consider a cake recipe an algorithm, because if you follow the steps and you have all the ingredients, you'll end up with a cake. In computing, algorithms provide computers with successive guide of completing actions and ultimately getting to the answer you need. So how does a probabilistic matching algorithm work? That's a very big word. We calculate the probability of a donor being a match, especially if all the information about either the patient or the donor is not available. And we try to do that as accurately as possible by using haplotype frequency sets. Those frequency sets are calculated on a population-specific basis. And so where do we get that population-specific information? Well, we get that from the global database because it covers the global population. Okay, so how do we calculate a haplotype frequency set? So a haplotype frequency set, or an HF set, is defined as the proportion of chromosomes of that type in a population. The WMDA uses these HF set configurations to better serve all patients, to provide an increased accuracy when selecting suitable donors and cord blood units for your patients. These frequency sets utilize high-resolution typing results from organizations to extrapolate the haplotypes of the region of that organization. So imagine a really, really large organization, let's say the US, the NMDP. It has an incredibly diverse population group. It doesn't really make sense to calculate a haplotype frequency set based on the US population because it is so varied. So but because the NMDP has really good high-quality data with enough of it, we can calculate population-specific haplotype frequency sets within the US population to improve the accuracy of the probability matching. So and that's what I describe here. Like, it's a geographically specific haplotype frequency set, and as long as you match the threshold of the minimum number of donors that you could potentially have for us to accurately calculate this set, we can do so. Unfortunately, not everybody is in that fortunate position. Most small and emerging registries, they don't meet that threshold. Even if they're incredibly diverse population, we would then have to apply the global haplotype frequency set to their donor pool, which does mean it is going to affect the accuracy. It doesn't mean that the donors displayed are wrong. There are still a high probability of a match, but the percentages that I'll indicate shortly will be, you need to maybe just make an informed decision or have this in the back of your mind that it might not be as accurate. We're talking a few percentage points here. So this is the probabilistic matching algorithms in action. What you see there is the search results for a patient. In the top line is the patient details, and then 2 potential donors. And those are the results. You'll see percentage points there, and all of the percentages calculated is based on haplotype frequency sets. On the far left, you will find the overall match probability. So it gives you an idea of what is the overall probability of a donor having no mismatches, 1 mismatch, or 2 mismatches. Whereas the locus-specific probabilities give you an indication of either it's a full match at a specific locus, or there is some ambiguity. There is some ambiguities that need to be resolved in order for you to establish whether this donor is the most suitable donor for your patient. However, not all donors are fully typed. So how can you make an informed decision? And again, here, the algorithms help you out because they still calculate the probability even though it does not have the HLA information. So in this case, for the second donor, without even knowing the donor's HLA, it can predict that there's a 71% chance of the donor matching the patient at the C locus and a 63% chance of the donor matching the patient at the DQB1 locus. So now that we've gone through the really hardcore techie tech stuff, let's go to the more practical things, like how we leverage these technologies to find you the best donor. So in a practical sense, what do? You log into, you go to the WMDA website, you log in using your multi-factor authentication because it is incredibly important that we keep the data secure. If you're not used to multi-factor authentication, I suggest you speak with your DPO or your security officer. It's incredibly important for the WMDA that we make sure that all patient and donor data is always secure. Because we are based in Europe, we have to comply with GDPR, and by extension, many of our members are also required to comply with GDPR. That includes the SABMR. So once you have logged in, you go to the screen and you add your patient data. Again, there's an element of consent. You will be prompted in a little pop-up box asking you, do you have consent from the patient to enter their very clinical information into a website? Once you've done that, please enter the required fields. At the moment, it's only HLA A, B, and DR, and this is now for the patient. You can also add the optional class 2 fields, and then just indicate the type of search that you want. Do you want it a donor search, or do you want a cord blood search, and which algorithm would you like to use? Now, this is all a manual process, so you are physically typing in information from a piece of paper that's sitting on your desk. However, there is an element of error potentially there, and the WMDA recognizes this, and so we have built out an electronic or API connection straight from your local system to this web interface, where you can simply automatically transfer all of your patient data directly to Search and Match. So you say, oh, I need a global search for this patient of mine. There's a button somewhere on your screen. You hit that, and all of your patient information is sent to the search and match service. By the time you've logged in there, there's no typing in patient information because it will already be there for you. Essentially, like in a perfect scenario, you're not transcribing the patient information coming from the lab. If you get the lab, your patient's lab information in a digital form, you can import that into your local system. And from your local system, you are simply sending it automatically, digitally to the WMDA. Again, element of quality control there is very easy to set up audit trails within your own local system to make sure that the information is completely untampered all the way from lab to the WMDA search and match service. So when you're performing a search for a patient, you know you're actually using the right HLA and you're not accidentally running searches for a 101 when it was supposed to be an 1101. So once you've entered the information or submitted, automatically uploaded the information, you can initiate the search. The WMDA has two algorithms available in the search engine. The one is called HAP-eSearch and the other Atlas. And there is no limit to how many searches you can run at the moment. You can run a donor search and a cord blood search using both algorithms in parallel, and it won't care. You can tick all the boxes if you want, and you can just tick one box if that's what you need. And once you've done that, those are your results. That was very quick because it's just a few clicks, but in reality, depending on the complexity of the search, whether you have the maximum amount of patient data available. Of course, if the patient's not fully typed, the search is going to take longer because it takes a long while then to calculate all the probabilities. And also, if it's a very complex search, so let's say it's an uncommon phenotype, it is also going to take a little bit longer. But on average, a search shouldn't take more than 5 to 7 minutes to complete. So you'll see the results on this screen. You'll see on your screen because you're logged into the search and match service. However, again, there is an alternative. Without having ever to leave your local system, you have now uploaded the patient to the search and match service. There should then also be a button on your system that says, okay, I've uploaded the patient, please run the search and let me know when the search is complete. Once it's complete, you can automatically download all of your donor and cord blood results from search and match back to your local service. Again, no room for error. You are not transcribing or copy-pasting donors from the website and like trying to get it into a Word file or into an Excel file. You can simply get all of the results back to your local database. And I need to reiterate that this is donors from all over the world. One search, all the donors. So from search and match, it will just come straight to your local system. And this is again using the technology built out by the WMDA. So now how do you choose which donor? I can't tell you which donor is going to be the best donor. It's going to be on a case-by-case basis, and after all, you're the experts. You know who the best donor is. But what can I do to help you get to that answer faster? And so within our search and match service, we have built a couple of tools. The one is using various filters to narrow down the results based on transplant center preferences. So we all know that physicians, they have the best overall overview of the patient's clinical situation. And while, let's say, a CMV positive donor was great for the last patient, the next patient's going to need a CMV negative donor, or they always want the youngest male donor. And there is a really quick and easy way to filter down from, let's say, 17,000 results down to a few hundred using all of the filters available in the application. Here are all the filters available for donor results, and as you can see, you can filter by registry, so that's a place in the world, whether or not they're accredited. their accreditation status, which again is really important. If it is very important for your organization to only utilize products from WMDA members that have gone through an accreditation process, you can filter for it. There's also different match grades, where the mismatches are allowed, and so forth. These are all the filters available for cord blood units. So again, if you have a very, very specific unit in mind, it needs to be incredibly specifically in terms of TNC or CD34 value. You can select it here. You can even enter the minimum allowed TNC or the minimum allowed CD34 and really, really narrow down that search for you. Again, you will see there is a filter for cord blood bank accreditation status. And that is not just WMDA, that is also NetCord, FACT, AABB, and yeah, I think those are the ones. Yeah. Maybe some percussive maintenance. Ah, there we go. Ah. No? Oh, is it back? No. So I'm seeing something different on the screen.
Something different.
Yeah, there we go. Thank you. The other feature that we have is automatic updates. So if your patient is currently in the active status, meaning that you are actively looking for a donor, the WMDA will provide you with an automatic refresh of that search every 48 hours. So you don't have to go and refresh to see if there's anything new in the world. WMDA will do this for you automatically and also signal which donors have either since been deleted or been newly added to any one of these searches for you. So in your patient list, you'll be able to see which patients are marked for automatic refresh in the column indicated there. And once the automatic refresh has taken place and you go back into that search, you will get a little icon. If it's a green circle with a plus in it, it means that that is a donor that you have not seen before for this patient. This is a new donor since you last entered this search. But conversely, the second donor on this list, this donor is no longer listed in the global database. So if you were considering this donor for your patient, don't. You can't anymore. Other features include grouping and sorting. So in searches for patients with pretty common phenotypes, it is not uncommon to get search results of 17 to maybe 30, maybe even 50,000 results. And you can narrow that down using various filters, but then at some point you also just want to know, okay, so which of these really have the best probability of being a match? And because they're all maybe a 10 out of 10 or a potential 10 out of 10, and then you can filter by the sum of probabilities. So it looks at the overall match probability in the column all the way to the left, and it would then sort them to the highest to the top. And so the one that is then absolutely at the top, while they might not be a full match, it is going to be the one with the highest sum of probabilities of being matched for your patient. And so with one click, one sorting tool, you can automatically just re-rank all of the donors and get the top one, the most suitable one, I guess, at the top. This is also possible for cord blood units. And then lastly, one of the features I'd like to highlight is setting user or transplant-specific preferences for search defaults. Now, it might not seem like a really valuable feature to have, but there is a very good chance that you have quality protocols at your organization that state that all searches need to happen in a very specific way. Now, instead of having to go and set all of those filters for every individual search that you run, you can all set them in your profile as the default. So you don't have to think about it again. You know that all of your searches in future will then comply with your quality protocol in terms of donor selection, all searches that you run in Search and Match. So if you're logged into Search and Match service, you can go there to the top and click on Preferences. And then the first list of options you'll find is in your patient list. Which tabs do you want visible? Do you only want to see My Patients tab, or do you want to see only the tab with all of the organization's patients in it, or all the tabs? And then also, what is the default visible tab? And so this is what it looks like. So just a quick explanation maybe. Everyone from the same organization will be able to not just see the patients that they've entered, they will also be able to see the patients entered by their colleagues within that organization. So let's say your colleague wins a trip to Hawaii and you need to take over all her patient cases for the next, let's just give her a week. Then you can just say, you don't have to reassign your patients to me. I'll just go and have a look at your patient cases in the My Organization's Patients tab. The other search defaults you can set is for search. So there is which matching algorithm you would like to use as default. So there's HAP-E versus Atlas. They are pretty much the same, however, they have been developed from slightly different approaches. So they are going to perform slightly different depending on the complexity of the search and depending on what it is you're looking for. If it's maybe multiple mismatched donors, you would prefer to use one algorithm over the other. Alternatively, within your organization, you have written a quality protocol that states that at this organization, we will only use algorithm X for all of our searches. And now, if that is the case, you can set it once and you don't have to go back and set it every time for every individual search. You can also set whether all your searches should be run default as a 10 of 10 or as an 8 of 8. Not everywhere in the world the match grade is exactly the same. Again, you're the experts, you already know this. But if you live in a country or you're part of a transplant center who match 10 of 10, then you can set that as a default. However, if you're only interested in matching 8 of 8, set it as a default and you don't have to think about it ever again. And the same goes for cord blood units. And then in terms of search results, what is it that you prefer to see? So what is the default sorting for mismatches? Do you want the standard sorting, or do you want the sum of probability sorting, where we will always rank the donor with the highest probability, overall match probability to the top? If you don't want to think about it again, just set it as a default and you don't have to apply that filter every time. Also, the secondary sorting is interesting because we're seeing maybe a shift in some organizations towards not really considering TNC as much for cord blood unit selection, but instead CD34. So if you don't want to necessarily have your cord blood units ranked by TNC, but rather by CD34, you can also set that as a default. And that's just a little bit of how that would look like in the screen for you. So what does the future hold for Search and Match? This is the realm of technology, and obviously, clinically, we're always trying to stay on trend, but it's mostly catching up with what all the brilliant scientists are doing out there. So firstly, there is a new product coming up. You already know donors, adult donors. You know cord blood units, and next up within the next 6 to 8 months, WMDA will start listing adult donor cryopreserved units. There might still be a name change, but this would be a product derived from an adult donor, cryopreserved and stored, like an, if you like, off-the-shelf product. There is also a new product feature called extensively tested cord blood units, which will include post-thaw and verification typing data. And so we will make a little picture or an icon that you can filter against in Search and Match. If you're only interested in seeing the cord blood units that have been extensively tested, that include the post-thaw data as well as verification typing to speed up your selection process, you will be able to sort for that in the not-so-distant future. And then lastly, Match Connect, which is sort of like my baby. It is the new registry-to-registry communication solution. Registries can send requests automatically after identifying suitable stem cell sources anywhere in the world. And then you can place requests, donor or cord blood unit related requests for those units using this service similar to what you will be using MDIS like today. So it is really fast, reliable, secure, and automatic. And so with that, I thank you for your time, and I hope to see you all log into Search and Match really soon.
Dr. Cochrane, if you can come up and then just address stem cells and challenges and emerging issues. So thank you very much.
Good morning, ladies and gentlemen, and I would like to thank the organizers of this conference for this wonderful opportunity to speak to you today. So I will be, as you can see, speaking about the challenges and emerging issues in stem cell processing. As you can see, I brought old age because apparently my size of handwriting didn't increase with my deterioration in eyesight, so I do apologize putting my glasses on and off. Today I will give you a quick background, and I know the majority of you know all about this. I will tell you about challenges that we experience in the laboratory, emerging issues, what is new, what's happening in South Africa at the present moment, and then a conclusion. I have no conflict of interest to declare. So as you all know, this is very common knowledge for everybody. Stem cells are undifferentiated, and they can divide in an unlimited basis, or they can differentiate into all of the different cell types. That is why it makes them such a fantastic type of cell to work with when we look at gene therapy or any other cellular therapies. So regenerative medicine is a very exciting type of medicine. And the main aim of regenerative medicine is to provide an alternative for a cell that is at the moment not functioning optimally due to either disease or trauma of the cell. And by replacing the cell or by changing the cell to deliver an optimal function. Now, part of regenerative medicine is advanced therapeutic medicinal products called ATMPs, and these are products that are mainly focused on a new product where our traditional products were ineffective. So they are focusing to specifically target diseases that we were previously not able to treat. They can be divided in gene-based therapeutic products, tissue engineering, and somatic cell-based products. They can differ from a single injection to actually insertion of a graft. They are a variety of products, but they do form part of the regenerative medicine area. Now, as you all know, neurological disorders can be treated with stem cells, something like Alzheimer's, Parkinson's disease, spinal cord injuries. We can actually either implant neural cells in them, or we can assist the ones that's there to be stabilized. Autoimmune diseases can be modified, preventing further deterioration, or otherwise just helping alleviate the symptoms. Research in like multiple sclerosis, systemic lupus erythematosus, type 1 diabetes are undergoing. We all know about the Geneva patient, which were cured using the CCR5 Delta 32 negative donor, and these are areas in infectious disease that are presently being investigated using stem cells. Now, we are very aware of the involvement of stem cells in oncology treatments, and I'm thinking of things like hematopoietic stem cells, CAR Ts, CAR NKs. These are all involved in oncology treatments. And then, of course, stem cells can then definitely be used when we look at drug discovery and development. So we can use stem cells to mimic what's happening in the body and therefore help us identify drugs and get a heads up in this, reducing the time of drug development and enhancing the development of new drugs for specific conditions. Stem cells are often used in skin grafts and other surgeries to increase the time that the patient takes to actually get better. So why am I talking today? So often the area that can be overlooked, because we often speak about who is the right donor, who is the best recipient of this donor material. But we often forget that there's a step in the middle. Somehow, by magic, the stem cells that's collected must be processed in a way in which the stem cells can then be given to the recipient. And in this processing step, that is where the laboratory plays a major, major role. So the laboratory are required to cultivate, to expand, to manipulate, to extend the stem cells and make sure that they maintain their integrity, they remain the type of stem cell you want, they didn't differentiate into something else through these processes. They must also be stable, and they must be accessible to when you need it, as you need them. The laboratory must ensure the safety of the stem cells as well as the efficacy thereof, and this is often used in our release criteria. This can vary from viability of the cells, the cell concentration. Can the cells differentiate into what other type of cells? So these are all things that we need to ensure when we make the cells or when we process the cells, that they will be sufficient for when we're actually going to reinfuse them. The laboratory can assist in advanced understanding of stem cell biology as well as the mechanisms of action. This can be done through looking at the stem cells following infusion and after processing. It's also extremely important to adhere to all regulatory and ethical guidelines when we look at our stem cell processing. I want to just remind you or tell you, for those of you who don't know, that there are a difference between minimally manipulated cells and more than minimally manipulated cells. The reason why I want to make sure that everybody understands that there's a difference, because there's a difference in regulatory requirements, there's a difference in the laboratory requirements in terms of the facility that you need to use them, as well as how the people will work with them. So an educational requirement from the staff. So minimally manipulated cells, they are normally just like concentrated, washed, a cryoprotective added, making sure they are viable, making sure they are at the right concentration. And one of the examples here are your hematopoietic stem cells. When we look at more than minimally manipulated, gosh, that's quite a tongue tire, more than minimally manipulated stem cells, normally here we use a mechanical intervention, a chemical enzymatic intervention, or we can expand these cells. So these cells, like your mesenchymal stromal cells, your CAR-Ts, your CAR-NKs, they require different regulations. They require different facilities. They also require a different way in which you work with them. Your workflow is different from the other. It's more stringent when you work with these cells. You must adhere to GMP. You cannot get away with that. And you must adhere to the regulatory requirements. They do differ between the two different stem cells. And by the way, that is mesenchymal stromal cells in the picture down there. Very nice one. So let's look at the challenges. So obviously, there's challenges everywhere, and in the laboratory we also have challenges. So firstly, are the cells viable? Did we get them viable? Were they transported, and did we receive them as a viable cell sample? After we processed them, are they still viable? After we cryopreserved them, did we lose any viability? Did we lose any cell concentration? Because as you all know, once we process and we work with cells, we will evidently lose concentration, but do we still have sufficient cells at a sufficient viability to ensure proper treatment? Are our culturing of our cells optimal? Do we have standard protocols? Are these standardized throughout our facility, within our country, between other facilities, internationally? What is the regulatory requirements? What is the guidance that our international partners give us in this? I'm just going to touch on quality control because we've got a fantastic speaker after me talking to us about quality control. But we need to ensure that we have quality control steps in every step of our process to make sure that what we are producing is at the right level of safety and at the right quality. This is a very practical example, storage. Anybody in a laboratory that works with stem cells will know that the storage of stem cells is a problem. You never have enough. Really, you never have enough. We process the stem cells. They are such unique, special, valuable units. We don't want to throw them away, firstly. We always think they might still be useful. The other thing is, if you don't have informed consent to get rid of your stem cells, you cannot get rid of your stem cells unless you have consent from either a family member, the recipient, the donor, whoever it might be. So our informed consent must include, can we dispose of these stem cells? When can we dispose of these stem cells? The ideal would be after a specific amount of time period, the donor no longer require it, these stem cells could probably go into a public bank. In South Africa, that is not a possibility because we don't have a public bank. So how can we, so in the end, we just store more and more and more and more of these stem cells, filling up multiple liquid nitrogen freezers, and we're hoping that they will still be viable or be usable one day. Another big problem is positive sterility. During your processing, if there's a sterility that is positive, it affects your product negatively. This product can at times be unutilized, cannot be used for transplant, and how do you go about this? So this is a major problem, and therefore the way in which we process our stem cells and our environment of processing our stem cells must be very clean, and our clean rooms must be perfect, and we must make sure that everything is working well. Disasters. I want to just, in South Africa, we had several of these in the fairly recent future. So let's think of disasters. During the outbreak of COVID-19, we could not always receive our reagent that come from international suppliers. So now we've got no reagents to process our stem cells. What do we do? If we have a flood, either our donors' stem cells can't get to the processing laboratory, or our staff can't access our processing laboratory, or the stem cells cannot be transported to the recipient. We are desperately waiting for these. So these are things that we have to take into consideration when it comes to disasters. In South Africa, we also have other man-made disasters, such as protest actions, which we also have to think about. What is our business continuity, our plan B? What are we going to do when this happens? Do we have another facility? Do we have a partner? Do we have a buddy that we can go and say, please, can I just process in your facility up until the time that I can get back into my own? What is our plan B? And as you know, this processing of stem cells require very specialized equipment. What if these fails? If your HVAC system fails, it means our clean room are not sterile any longer. That can negatively affect the quality of our stem cells. Our equipment failure can be a really big problem when we think of stem cells. If a liquid nitrogen freezer fails, you have multiple units of stem cells in that freezer. That can negatively affect the viability of those stem cells in the freezer. I know one of our biggest problems in the facility that I work for, by the way, is the South African National Blood Services, is that if we have a problem, our first thing is our liquid nitrogen freezers okay? If there's a problem with the delivery of liquid nitrogen because of the trucks being in, there's a protest action or there's whatever. Our liquid nitrogen cannot get to our laboratory to fill up our liquid nitrogen tank. How do we keep our freezers at the optimal level to make sure that we can actually maintain the viability of our stem cells? These are things that obviously is our problem, not the clinicians' problems, but these are things that we think about when we think about our business continuity and what can we do to ensure that our processing of stem cells are at the right expected quality. So obviously it's also ethical and regulatory compliance. Regulatory compliance is SAPRA compliance, SANAS compliance, and for the international accreditation units, JC compliance. Ethical, do you have consent? Do you have informed consent? Was the informed consent obtained in the right way? When we look at new stem cell therapies, we often start with a small proof of concept study. However, this is often not at therapeutic levels in terms of cell concentrations. To upscale this, you need to make sure that your protocols are robust and cost-effective. We also think of the long-term safety and efficacy of our products. Then, laboratories must keep abreast with technological advancements and innovation. This will ensure the suitability of your product and ensure that we are actually at international standards when it comes to our stem cell products. So let's just talk about additional challenges in South Africa. So firstly, the South African healthcare system, there's a large disparity between private and public healthcare at the moment. Therefore, there's also a large disparity in terms of the laboratory, public versus private. The public health sector at the moment is dependent on processing facilities such as the South African National Blood Service for the processing of their hematopoietic stem cells, while the private facility has more options. When we think of human resources, we do not have sufficient technologists, scientists that are actually equipped to handle stem cells, to manipulate stem cells. Yes, we train them. It does take time, though. So we do have them, but they are very few. The regulatory framework is often lacking, and there's ethical considerations to be taken into consideration. And when I say the regulatory framework is lacking, I will give you an example that colleagues of ours in the Albert Cellular Therapy Laboratory recently experienced. They had to, for their viral vectors that they want to use for the CAR T cell, they had to get veterinary approval from the Veterinary Regulatory Office and not the Health Office. So these are nuances that often has to be navigated. Obviously, funding is limited. In a country with a high communicable and non-communicable disease burden, to motivate for funding for stem cell therapy that can probably only advantage a small number of people can be quite difficult. So resources are limited, and the funding opportunities are sometimes limited. And therefore, it's very important to have collaborations, because together we are strong. And networking with local and international partners assists in overcoming a lot of these challenges. So let's talk about what's new, emerging issues. We all know that cellular therapy evolves quickly, and that genetic manipulation and genetic therapies evolved just as quickly. We have silencing of genes, we have expression of other genes using gene editing technologies, and one of the very interesting ones is CRISPR-Cas9. For those of you who know about it but not really know about it, CRISPR-Cas9 has the ability to cut double-stranded DNA at a very specific site, allowing you to either insert genetic material into that genome or delete material out of the genome. By doing that, you have targeted genetic expression. And through protocol improvements, off-target expression has also been reduced. When we look at viral and non-viral vectors such as the adeno-associated viruses, can be used because they target very specific cells. So if you add a genomic sequence in a viral vector, such as these AAV virals, they can then be transported to a specific cell. Unfortunately, their load capacity is low, and that is where non-viral vectors come in. They have a far higher load capacity, they are safer to use, they are easier to process, and they need less stringent environmental controls and laboratory infrastructure. So in my opinion, the emergence of non-viral vectors in gene editing and gene technologies will increase rapidly in future. Another interesting field that has emerged is inducible pluripotent stem cells. We all know that there's a major ethical concern when we think of embryonic stem cell research. This was to a large extent overcome by inducible pluripotent stem cells. What these are is it's somatic adult cells which has been reprogrammed to be embryonic stem cell-like stem cells. This assists in the investigation of research through either personalized medicine. We can actually take a person's somatic cells, adult cells, and regenerate them to look at the disease progression. So there's a lot of research going on in iPSCs, and there's a lot of potential for these cells. Some other advances are the advancement of three-dimensional cell cultures. These use scaffolds, and they actually then mimic what is happening in the body. They can look at the interaction of these cells with other cells in the area, with the extracellular matrix. Now, organoids are like small organs, and this is often used for disease progression and drug therapy and drug interactions as well. When we look at microfluidics, that is the picture on the right. Microfluidics has the ability to, in a very controlled way, monitor and manipulate the environment in which the cells grow. This is very small little discs, so we have a lot of cells in a small area, which increases the time that it takes to do research. Nanoparticles can be programmed to attach to a very specific cell or tissue, and therefore you can have targeted drug delivery to that specific cell or tissue. While automation, obviously, as always, reduces human error, increases your speed in which we do things, and your accuracy. High throughput screening, as you can see below, the picture below, this is often done where you have like a different agents that you want to screen, or you have a different matrix in which you want to screen, and through high throughput screening, you can then manipulate the environment in which these cells grow to determine what the best option is. The analysis of data via high throughput screening can be quite challenging, as you can imagine. So we also, when in the laboratory, we have to make sure that we're abreast with the latest international guidelines to ensure that we stay relevant and that we do the best possible way in which we produce our cells. There's an international shift to use serum-free everything, serum-free medias, and to be able to do this, we have to think of alternative ways in which we have either our growth factors or anything else. High contrast screening technologies is a very high powerful microscope with a robotic arm that can automatically focus on the cells. It can then determine the cells and extract the data. Again, the analysis of these can be quite difficult, but that is one of the main cell-based assays that's also used during screening processes of stem cells. Now, a talk about new options can never be a talk about what's happening without artificial intelligence. So we know artificial intelligence will be very important in regenerative medicine because it empowers researchers to analyze vast amount of data. It recognizes patterns which often the human brain cannot recognize or will take a long time to recognize. It can make predictions based on data that it has either already acquired or that has been imported into the system. Just to remind you, artificial intelligence have developed through time. Originally, we just called it artificial intelligence, which mimicked the human behavior. That involved into machine learning, which is techniques that can improve the tasks without explicit programming, and now to deep learning, which are algorithms that adapt and learn from a vast amount of data, to the point that the analysis of these data the computer itself or the algorithms can determine the type of cell and then adjust accordingly without human intervention. We get computational simulations. We can also get drug development. So AI can actually try and identify new targets in our drug by simulating the disease. It can model the disease, trying to think of new ways in which we can approach it, which way in which we can treat it. It has predictive modeling abilities. It's also very important in personalized medicine. Tissue engineering, as I mentioned previously, are scaffolds that we need for our three-dimensional cultures. These needs to be able to support the growth of the cells. So AI can then assist us to determine what is the right type of scaffold that needs to be used. And in cellular therapy, it can determine the best cell to use for a disease and even indicate to the laboratory people what is the best condition to grow this cell in. Now, what's happening in South Africa? So presently, we have a variety of hematopoietic stem cell transplant centers within South Africa. These are both throughout the public as well as the private sector. Unfortunately, our South African Association of Standards does not accredit stem cells. So what happened is that there's 2 facilities in South Africa that underwent the international JC accreditation, similar to FACTS, and this is the South African National Blood Services as well as Idrc Cellular Therapy. The South African National Blood Services offers a mobile collection unit as well as a central processing facility, and it services both the public as well as the private health sectors. The Idrc Cellular Therapy services the private sector. Now, one of the very exciting areas in the country is the potential of CAR T cells, as you have heard earlier, and a safety trial is going to be conducted by the Idrc Cellular Therapy as well as OncuLab. So we're very excited about the future of stem cells in South Africa. So just to conclude, obviously, as always, there are numerous challenges, but there are ways to overcome these challenges. Technology is fast developing, and we have to keep abreast to ensure the best safety for our patients and that the best potential way in which we process our cells is there. Collaboration is essential, especially in a resource-limited country such as South Africa. And there's a promising future in stem cells that will transform healthcare as we know it and the well-being of our patients. So thank you.
Just to say, Brianna, I forgot to mention that you are an excellent teacher and you're well known. You're a PhD medical scientist, and she heads up the research division? Translational. It's called Translational Research and Stem Cell Therapy Unit. Well, it's not a therapy unit yet, but stem cell in research as well in the South African National Blood Service, which is sometimes referred to as SAMBUS. And I think that brings us to our last talk today, which is around the quality one. Hirosine, if you could come up, please, and address the audience.
Hello, everyone, and thank you for the committee to giving me the opportunity to present today. And disclaimer, I'm not a lab scientist or a processing expert, so my view is just from a quality perspective. And it's a good thing that, Rihanna, you've set the scene by going through all the processes from a laboratory perspective. And I'll start my presentation with a short story. So in 2020, when the pandemic started, and we were all working remotely, and there were a lot of changes happening that we needed to react to the changes. And the first thing, one morning, I received an email that, there was a World Cup already. We were getting ready to import stem cells, but the only way for us to bring the cells into the country was for the cells to be cryopreserved before importing them. And that was something new to us that we've never done before, because normally processing is done before COVID, it was routine for processing cryopreservation to be done by the collections, the transplant centers. So once the cells have been collected, they're shipped, delivered, and then the transplant center will do the cryopreservation. So because it was a first for us, we needed guidance from the competent authority just to check if it was okay for us to get the processing facility to cryopreserve the cells before they're shipped into the UK. But at the time, I was acting as a designated individual for our regulatory license, so I needed to make that contact with the competent authority just to check with them what process we needed to follow. And the advice I got was, you need to assess the processing facility to make sure that they meet the UK requirements. So you need to assess, the way they process the cell. You need to give us assurance that the quality and safety of the cells will meet the UK requirement. And I said, okay, I can do that. So just let me know what I need to do, how I need to do that assessment, and then I will get started because we don't have time. But they said, oh no, you need to tell us how you're going to do it. You need to decide how you're going to do that assessment, and then we will review it and let you know what we think. So basically, you need to assess the processing facility, and then we're going to assess your assessment and make a decision. Phew. So I did consider running away at that time for a split second, but unfortunately all the borders were closed. And obviously there was a patient waiting for treatment, so we needed to act fast and then put that patient first. So that just outlined one of the challenges, of like cross-border compliance, meeting like the regulatory compliance when you're importing, when you're exporting. So my presentation today, I'm just going to outline like the issues that we face from a quality perspective, like what are the challenges and emerging issues when it comes to stem cell processing. So I'm not going to get much into, what is processing, how it's done, because the expert has already gone through it. And my presentation could basically just be everything that Rihanna's presented. Behind every step of what is done inside the laboratory, there's someone, it could be a quality manager or someone who oversees quality. So their role is to make sure that every step of the processing within the laboratory is done in line with the regulatory accreditation quality standard to make sure that the processes are followed. So this is just a broad picture of the current applications, the journey of the cells. This is from like the Anthony Nolan register, how it's done. So the normal procedure for transplant, a patient is in need of stem cell transplant. A donor is found for that patient. A donor is matched, and then the donor goes through the donation journey. They attend the collection centers. The cells are collected. Everything is done to check the quality of the cells, and then if cryopreservation is required, this could be done at the collection center or at the transplant center, and then the cells are shipped to the transplant center for the patient to have their treatment. And then there's also a cell and gene therapy or research, so the journey of the cells. There's a client that requests cells, and then there's a donor that is also found, and they go through the donation, they attend the collection center, they donate, and then the quality of the cells also checked. And then there could be cells enrichment, cells isolation. This is done, could be done by the collection center or our cell therapy center. And then if cryopreservation is required, this is done. The delivery cells are delivered. But sometimes the difference could lie in like testing requirement for transplant and testing requirement for cell and gene therapy. So from the regulatory compliance point of view, testing could be different. So if PBMC is required, then testing of that donor must be done on day 0, collection day, or within 7 days. But if it's for transplant, a donor is donating PBSC or bone marrow, so the requirement is for testing to be done within 30 days of collection. So when the donor attends the medical, blood is drawn and testing is done. So obviously the landscape is changing because we're seeing, lots of changes from the normal regular transplant. We're seeing CAR-T treatment that is increasing. We're also seeing ATMPs being developed. So we know, we already have a feel, that in the coming years, things are going to be different. More changes are going to come because new advanced therapies, new treatments, new technologies are going to be created. And how do we prepare from a quality point of view to make sure that, compliance is always maintained, to make sure that patient safety is always at the heart of everything that we do. So some of the challenges that my predecessor have already outlined is just how to navigate the regulatory landscape. So I've just mentioned in my story that, if you're importing from outside of the UK, or for them, they export it. So when that exchange that is happening, we need to make sure that from the two sides of the requesting and then the donor where the cells are coming, that from both sides, compliance is maintained. We need to make sure that the quality and the safety of the cells are maintained. But it's always a challenge, even within one country, to navigate, between different regulatory bodies, and it makes it even more challenges when it's cross-border. So making sure that quality is maintained throughout the journey of the cells. And also there's a quality control. So how do we make sure that the integrity, the quality of the cells is also maintained throughout the journey? And then how do we also make sure that across the world, like, cross-border processes are standardized so that to maintain consistency and also like the procedures that we use are reliable. We get the same results every time that, the cells go through processing. And if you're moving from a small lab clinical production to a small lab processing, and then you have to move to a wider clinical production, how do we make sure that compliance is also maintained through that transition, making sure that, GMP compliance is put into consideration, making sure that all the steps are put in place for the quality and safety of the cells. And we also have emerging issues. We have new technologies coming up. We have also, like, we know with cell and gene therapies, we know that ethical concerns is not going to go away. So we need to make sure that, we protect the safety of the donors. We need to make sure that the donors are involved in that decision, in that journey that we're making with new technologies, with cell and gene therapy. We need to make sure that the right consent process, what is in place to make sure that every donor knows what they're consenting into and what their cells are going to be used for. And with development of new biomarkers, we need to make sure that quality is also taken into consideration. So how do we make sure, how could we ensure that with all the challenges and all the issues that are emerging, how do we make sure that, from a quality perspective, that regulatory compliance is at the center of what we do? So we have to make sure that we maintain all the standards. Everything that we do, every step of the process is done to the highest standard. We have to make sure that, the processes that we're developing are safe to use and are also effective, and the final, the outcome, because there's always that patient at the end of everything that we do. So patient safety should be the focus of every single step of stem cell processing. And I mentioned ethical responsibility earlier. So how do we make sure that everything that we do, we're being responsible, the way we're using the cells, the way we're sourcing the cells, so from collection, processing, the way the cells are going to be used, how do we make sure that ethical responsibility is always there? And also how we need to pave the way from, regulatory compliance for border acceptance. So when we're exchanging cells, across borders, we need to make sure that compliance from each country is also taken into account. We need to make sure that there's always a way to ensure that the patient that is receiving the cells is satisfied that, the product that they're getting is going to cure, that is going to help them, that, the product will be safe to use. And we need to also make sure that there's always measures in place so that every time there's a consistent result to, like, the product always comes out how we predict it to come to be processed. So the final product is always of the highest quality to be used. So mentioning the cross-border compliance, an example is for the UK. So let's say if we are importing cells into the UK, but the cells need to be processed before we can import them. So there's a process in place for assessing compliance of the processing facility, and from, the facilities, the equipment that is used, the reagent, all the personnel that are involved in that procedure, the processing, making sure that they're qualified, they're properly trained, and also if the processing facility is accredited, an example of fact accredited, that gives a layer of assurance that they adhere to international standards. So there's an example also is like environmental monitoring. So how do we make sure that the condition in which the cells are processed are safe, to ensure that the product that would come out of it is also safe to use. So following that assessment, if we found that not all the UK requirements are met by the processing facility, obviously the final decision is not Anthony Nolan's to make, but we pass it on to the requesting center, the transplant center or the client, and it's up to them to make a risk assessment to review all the gaps, all the requirements that are not met by the processing facility, and then make a decision on whether they still want to go ahead and for the cells to be released under clinical concession. So there's also inspections by the regulatory bodies and accreditation bodies. So these are important because they would come in frequently to assess all the processes, assess all the steps that are taken into consideration when manipulating cells, and they want to make sure that the facilities are fit for purpose. They want to make sure that compliance is maintained throughout the process. So having inspections regularly is a way that we could guarantee that some of the challenges could be addressed by all the facilities that take care of cells processing. So this is, I'm not going to go through like every single step of this. This is just an example of a workflow at the Anthony Nolan Cell Therapy Center. So this is how a workflow is set every day for cell processing. So in each step of this workflow, it's just making sure that they think of like the quality standard, what is required, what do we need to take into consideration, what do we need to follow to make sure that from starting this procedure of process and to the end result, the product that would come out of it. In each step of this workflow, they want to make sure that quality is taken into account. They want to make sure that compliance is maintained with each procedure. They want to make sure that the final product that would come out of it is safe for the patient to use, is safe to be released to the transplant center or the client. So this is just an overview also of processing criteria at the Anthony Nolan Cell Therapy Center. So for cord blood unit and acceptance criteria, it needs to be the unit from collection to when it's ready to be processed needs to be under 30 hours. And then, but for processing of stem cells from adult donors, the acceptance criteria is defined by the transplant centers when it's sent to the cell therapy center for processing. So they define the acceptance criteria. So when the cells are received, Anthony Nolan Cell Therapy Center will just go ahead and do the processing. So for our code, the Anthony Nolan code processing, we use the CEPAX2 system. And for HPC collected from adult donors. The cells are processed manually, so by using a grade A flow hood. And then cells are frozen at the Anthony Nolan cell CTC using control raised freezer before being stored into cryotank. So these are just, this is just an overview of, the testing, the quality and safety of the product. So in each step inside the cell therapy center, compliance, quality standards are always put into practice. And storage, like Rihanna mentioned, storage is also very important to make sure that the integrity of the cells is maintained for the whole duration of storage. And this is also a regulatory requirement for storage to be done in line with our regulatory standards. So in conclusion, we've seen how navigating the regulatory landscape is a challenge, and it's even harder when it comes to cross-border collaboration. And we also highlighted that there's still a need to keep working on, making sure that we meet the ethical requirement when it comes to research or other forms of treatment. And there are also new technologies emerging, but we need to make sure that quality standards are also maintained. So quality needs to be at the heart of everything we do. So when thinking of developing new treatment, new technologies, when thinking of switching, updating processes, we need to think of what the impact would be on the quality and safety of the product, what the impact will be, how this will go in line with all the compliance, the regulatory requirements that are already set in place, and also how could we shape the way regulatory requirements are set. So do we have an influence? Could we also have a part in making sure that whenever new standards are developed, new standards are written, we take into consideration emerging issues, we take into consideration new treatment, new technologies that are being developed. So it's really important that we make sure that quality is embedded in every step, every decision that we make when it comes to cell processing. And then thinking, always putting the patient first, always putting, the safety of the patient will always remind us on why quality is important. So quality is part of everything that we do. Quality is at the center of every decision that we make. So whether it's operational or whether it's inside the lab, quality should be the main focus. So making sure that everything that we do is for the safety of the patient at the receiving end, the patient that is awaiting their treatment. We have to make sure that the product that we're developing is safe for their use. So that was it for me. Thank you very much for that.
Thank you, Hirosine, for that lovely talk, and also finishing off with a good message as we develop all these new cell therapies and we move into a new era, possibly, is to not forget the quality and the patient safety. At the end of the day, that's what it's all about. It's about the patient. I think we started slightly late, but if I could delay questions, if you can catch people, our speakers who've done such a wonderful job at lunchtime, then I will let you go, unless there's one or two important questions that you're burning to ask. Well, you will find the speakers, and I've just got something to say to all of you. Thank you so much. Thank you for reminding us of where we're going. And thank you for bringing the quality aspect into it. And I just want to give you each a little gift for having done it.